I am Lee Gilman. My pronouns are they, them, and I am an assistant professor at Kent State University, which is located in Ohio in the United States. I am a neuroscientist by training, and my overall research studies how stress and diet can interact to influence brain health and behavior. So the talk that I'm giving you all today is looking through the lens of how dietary salt can elicit or exert these sex-specific effects on indicators of neurodegeneration risk. And I just have to state that I have no actual perceived or potential conflicts of interest, so nothing to disclose. I do want to make a land acknowledgement stating that the traditional people that this land belongs to, where my lab is located, include the nations of the Delaware, Miami, Mingo, Ottawa, Shawnee, Wyandot, and Cayuga. This land acknowledgement pays respect to the traditions, ancestors, and contemporary cultural and spiritual practices of Indigenous Americans. I ask you to please take a moment to consider the legacies of violence and displacement that have removed Indigenous Americans from their land. My lab, the Gilman Lab, we recognize that our work takes place on this land that was forcibly and unlawfully taken from these Indigenous nations. So I'm going to first cover the format of this talk, the information that I was provided, and set up expectations for this talk. So this webinar is part of an overall topic covering the role that lifestyle and environmental factors play in the progression of neurodegenerative diseases. I was told that the audience for this talk would be researchers in academia and industry, grad students, and postdocs. So I try to not go too much into jargon or very complex explanations. And I was told that the duration for this talk should be around the 25 to 30 minute range. So with that context in mind, I'm going to first start off discussing disease progressions, particularly as it relates to neurodegeneration. So if we consider the lifespan of an individual, historically the study of neurodegenerative diseases have focused more on people who are older, specifically the elderly, late in their life stages. And consequently, a lot of research has focused on trying to mitigate or prevent, attenuate early indicators of neurodegenerative diseases as people reach those stages where those symptoms become more apparent, right? Mild cognitive impairment, hand tremors, etc. But the way that I view things, I consider that neurodegenerative diseases really span the lifetime from conception and in utero all the way through adolescence, early and middle adulthood, into this later age if people are fortunate enough to reach old age. So my research is more focused on this middle area of early and middle adulthood, what components, what factors can influence brain health, particularly through the context of neurodegenerative diseases here. So when we consider this lifespan, there are things like stress that are known to influence the progression of neurodegenerative diseases. These stressors can be very acute, right? One-time, potentially intensive stressors, even traumatic, or stressors can be more periodic here and there, relatively mild, but still stressful experiences like having to stand in line at the DMV, right? Plus day-to-day, maybe even hour-to-hour stresses, worrying about money. Can I pay the bills? What do I have to budget for, right? When's my next paycheck? Or, I just got fired. What am I going to do to support myself, the people that rely on me to care for them? These are all components of stress, right? In its many varied forms. And there is a lot of research showing, broadly speaking, that stress can impact the progression of neurodegenerative diseases. So that's one factor that is well established in the progression of neurodegenerative diseases. Another factor that has been studied in the progression of these diseases is diet. How healthy a diet is has also been separately studied to evaluate how this influences progression of neurodegenerative diseases. And not just diet alone, but what people often might not consider is that there are also lots of condiments, right? Spices and sauces and dressings and things like that that people add to their main food components, right? And how do all of these aspects of diets influence disease progression? So generally, diet as a contributor has also been studied in the context of neurodegenerative disease risk. So we know that diet can influence this disease progression, and we know that stress can influence this disease progression. But there is far less research looking at the interaction of both stress and diet on markers of potential neurodegeneration risk. Stepping back a moment now from those individual factors, and if we just look at what has been found in the broad context of neurodegeneration risk across many neurodegenerative diseases like Alzheimer's, Parkinson's, Huntington's, multiple sclerosis, amyotrophic lateral sclerosis, right? All those different things. What are some common threads indicating that someone might be at higher or lower risk for neurodegeneration broadly? Those early indications, reviewed in these excellent articles here, these indicators can include oxidative stress, neuroinflammation, cerebrovascular disruptions, impairment in cognition, and more recently, type 2 diabetes. So each one of these has been identified individually as a measure that can indicate early on someone is at more or less risk for neurodegeneration. What do all of these have in common? From my own biased research lens, I see all of these and I think salt. This is what I study, one of my primary areas of research. Salt can affect all of these measures. Dietary salt, and all of these measures can indicate neurodegeneration risk. So my research that I'm going to cover for the rest of this talk, because we have relatively a small amount of time, I'm going to focus on neuroinflammation and cognitive impairment as early indicators of this risk through the context of looking at dietary salt and how it interacts with stress to influence measures of these two risk indicators. So why salt? So much research, dietary research, focuses on high-fat diets, different types of fat, high sugar or carbohydrate diets, or the combination of the two, high-fat, high-fat, high sucrose diets, which are considered Western diets. But not that much attention has been paid to salt in the diet and how this affects brain health. Far more research on salt in the diet focuses on cardiovascular measures and a little bit more recently, metabolic measures. But there is relatively little research on how salt affects the brain, right? Neural processes, neurophysiology. What interests me particularly about salt is that it has no calories. It's calorie-free, unlike fat, unlike sucrose, unlike carbs, unlike protein, right? But nevertheless, salt is able to enhance flavor. So it can help something perhaps bland, like a vegetable, make it more flavorful, so someone is more likely to eat it. And salt can enhance the flavor of something unhealthy, right? Like French fries, and result in potentially overeating of unhealthy foods. And like I mentioned, there is very little research on how salt affects brain health. And as a consequence, that means that there is not much studied in terms of how dietary salt impacts behavior that is regulated by the brain. Of those relatively few behavioral studies on the effects of salt in the diet, hardly any of those include biological sex comparisons. Most of them only use males. And for this talk, I am referring purely to biological sex. I am not making any statements about gender. So for this talk, I'm going to cover data that we have generated to test two overall hypotheses. These hypotheses, for the first part of this talk, I'm going to discuss data that we have gathered trying to address the hypothesis that acute stress after a long-term high-salt diet will increase neuroinflammation. And then second part of my talk, I'm going to discuss a different project where we tested the hypothesis that eating excess salt would reduce performance of a learned stressful cognitive task. So both of these were looking at stress and salt and how their interaction affects a metric of neurodegenerative disease risk early on, neuroinflammation or cognition. Again, looking at the interaction between salt and stress. So the first hypothesis that we investigated, I'm going to go over the timeline or the general experimental setup. And this project was led by my former grad student and my very first grad student who just defended her PhD successfully in June of last year. So this project that Dr. Beaver led involved us using C57 Black 6J mice. And these are all adults when we use them. And at what we call week 0, we assign mice different diets. This is when their diet manipulation commences. What are the diets that we give them? Mice are assigned either to a low salt diet, which is 0.4% sodium chloride or salt, and a weight per weight percentage. This diet arrives dyed blue. Okay, so it's shown blue here. And this high salt diet arrives dyed red, okay? And this high salt diet includes 4% sodium chloride. And because it's dyed red and this low salt is dyed blue, the way that I'll be showing the data in a moment for this study, we use blue circles to indicate the low salt diet, and we use red triangles to indicate outcomes from the high salt diet mice. We have these mice that are either just consuming low salt or just consuming high salt starting at week 0, and they continue consuming these diets until week 8, okay? And at week 8, they experience a very acute 6-minute stressor. And then just after that stressor, we collect brain tissue and process it so that we can measure IBA1 positive cells, which you can see up here in green. These are IBA1 positive cells. This is a marker for activated microglia in the brain. And the number of IBA1 positive cells can be used as a proxy to indicate to us neuroinflammation occurring in the brain, where more IBA1 positive cells indicates more neuroinflammation. What did we see in her study? We looked at several different brain areas that are important for stress responses, but here I'm just going to focus on what we found in the medial prefrontal cortex, or MPFC, of these mice. So the medial prefrontal cortex is a brain region that's very important for things like regulating emotions, making plans, focusing attention, many aspects of what we call executive function. And so when we walk through the results here, we see on the y-axis, we have our IBA1 positive count. So the number of IBA1 positive cells in our region of interest, medial prefrontal cortex. And on the left side of the graph, we have females. On the right side of the graph, we have males. When we first look at our low salt condition, where the mice did not experience any stress, we see that they have similar levels of IBA1 positive cells in the MPFC. When we look at our high salt mice in both sexes that have not experienced stress, we again see similar levels of IBA1 positive cells in the MPFC. When we bring in our low salt stress mice, okay, so these were assigned low salt and they experienced that acute stressor after the 8-week diet period, we see that stress did not influence IBA1 positive counts in females, but in males, we see a significant increase IBA1 positive cells, indicating greater neuroinflammation in males on low salt diet that experienced stress versus males on low salt diet that did not experience stress. So we see, again, stress increases IBA1 positive cells in males. But when we combine high salt and stress now for both females and males, what we see in females is that when they are consuming high salt and experience a stressor, we now see a significant increase in IBA1 positive cells, okay? Whereas males, we do not see a change in their IBA1 positive cells. Contrasting with the low salt, this shows us that high salt in the males is actually preventing this increased marker for neuroinflammation that stress produced in the low salt males. So high salt appears to be preventing that increase in neuroinflammation, whereas in contrast, we see the combination of stress and high salt in the diet in females increases this marker for neuroinflammation, and that in these stressed high salt females, they're also exhibiting more of this marker for neuroinflammation than males that were stressed and on high salt. So, for an interim conclusion based on those data, for neuroinflammation specifically, we observe that this acute stress-induced increase in neuroinflammation is mitigated by high salt in males, whereas it is exacerbated by high salt in females. Returning to our hypothesis for this project, this first hypothesis, we thought that acute stress after that long-term high salt diet would increase neuroinflammation. What our results tell us is that the female data we found support this hypothesis. However, our male data do not support this hypothesis. In fact, we observed in males that the high salt diet actually prevented an increase in neuroinflammation that was induced by acute stress. Really interesting results. Moving on now to our second hypothesis of eating excess salt would reduce performance of learned stressful cognitive tasks. Okay. Here's how we investigated or tested that hypothesis. This project is currently being led by a grad student in my lab, Grace Sonic. So the timeline that we used here, I should note we are using Wistar rats here instead of mice. Okay. We start, as with the other project, we're in adult Wistar rats. We assigned their diet. And this project goes for 6 weeks instead of 8 weeks like with the mice. As with the mouse project, we have a low salt diet, the exact same 0.4%. We have other rats that are assigned to a high salt diet, 4% salt, again, blue and red. Okay. So for the upcoming graphs, blue will be indicating our low salt group with a solid line. Red triangles with a dotted line will be indicating our high salt only group. But for these rats, we brought in a third diet group. That third diet group, we wanted to find out, well, what happens if we give rats access to both low salt and high salt? Because if you think about it from a human perspective, no humans just eat one thing all the time. They can choose, they can change it up. So these rats in this condition have access to both 0.4% and 4%, or both low and high salt diet. And so we call this group our mixed diet. And this group is represented by purple squares. So those are the diet conditions for this experiment. But remember, we were looking at cognition here. So how do we evaluate cognition? After the rats are on diet for 2 weeks, we train them, which involves 5 consecutive days where the rats learn a stressful task. They get 30 trials each day to learn that stressful task. What they are learning is proactive avoidance behavior. So I'm going to take a moment to explain this. Rats are placed into a 2-chamber apparatus that has a door between the 2 chambers. Whenever the rat is on one side of the chamber, for each trial that occurs, 2 lights are going to go on in the chamber where the rat is currently not present. And what the rat needs to learn is every time these lights come on, as soon as they come on, it needs to move over to where the lights are so that it doesn't experience a mild but unpleasant foot shock on the side that it's currently residing. So it needs to learn, oh, the lights came on, I need to move over there so that I don't experience this unpleasant mild foot shock. And this is what we call negative reinforcement, and that can be kind of hard to wrap your head around, but we frame it as proactive avoidance. So if you think about general life situations, so for example, we pay our bills on time when we can so that we don't have to pay a late fee, right? We are doing something proactively avoiding a late fee by paying the bill on time. Or in a maybe more daily home example, perhaps your partner always complains because you leave the toilet seat up, right? So you start remembering, oh, I need to put the toilet seat down so that I don't hear complaints from my partner. Or we stay roughly within the speed limit when we're driving because we want to avoid getting a speeding ticket, right? Or we don't talk back to our supervisor so that we can keep our job and not get fired, right? All these daily occurrences, life occurrences where we have to proactively avoid something. But because we can't speak rat language, right? The rats have to learn this task without us being able to give them any instructions. So we measure over time how well they learn this across these 5 days. And then we wait 4 weeks until week 6. And then we test them for 3 more consecutive days to look at their memory of this proactive avoidance test. So when we look at their responses, every single day they get 30 trials. So for this graph, we're looking across time for training and then 4 weeks later at testing. And on our y-axis is the percent of 30 trials for every given day. So for example, on day 1, the rats underwent 30 trials. And on day 1, about 25% of those 30 trials, the females exhibited proactive behavior. But by training day 5, we see that females in the low salt group, because this is our blue low salt group, by day 5, about 80% of their 30 trials, they were engaging in proactive behavior. So we can see this learning. And then when we look here at the 4-week time point, we see that they remember how to perform that cognitive task pretty well, near 90, 95%. When we look at our high salt only group in contrast to our low salt, we see no differences in the behavior here. There's no significance here. And when we bring in our mixed group, we again see no differences. And while this might seem like it's disappointing, it's actually pretty important and interesting because it tells us that female performance in this stressful cognitive task that they have to learn and remember, dietary salt isn't affecting them at all, which is kind of good news, right? Potentially. So if we then look at males, and we look at, again, across time, they are learning. This is an expected biological sex difference that we typically see in rats. We see that eventually by day 5, the male rats are learning. And I do want to note that these studies are ongoing and our male ends are a little bit low, but for both of these groups, we're still underpowered because we haven't yet achieved the ends indicated necessary by our power analyses before we started this project. Nevertheless, the data that we currently have, we see that our low salt males are learning and that they remember pretty well, even though we get a little dip in performance here. Now, when we look at high salt only males, we see that they learn similarly to the low salts, but we find up here that on the second and third testing days, that high salt males are actually performing better than low salt males, which is what's indicated by these little diamond symbols here. And then when we bring in the mixed males, there are no differences between the mixed rats and the low salt rats. So we're only seeing this significant difference between high salt rats and low salt rats only in males. So high salt is helping the males perform better and they remember this task. So it's enhanced by high salt. As we were running this experiment, we also wanted to assess food consumption in these same rats that underwent this stressful learning task. We wanted to see how does their food consumption change over time. When we look here, we're again looking at time on the x-axis, but now we're looking at weeks 2 through week 6, and we're just looking at their food consumption, specifically how much food did they eat per day, and we've normalized this to a percentage of their body weight because adult male rats are just naturally much larger than adult female rats. When we look at food consumption after this stress, so the stress bar indicates those 5 consecutive days where they were learning that cognitive task of avoidance, we see there's actually no differences in their food consumption in the low salt group. We similarly see no differences in food consumption in the high salt group. And when we look at total food consumption in our mixed group, we again see no differences. We were hypothesizing we would see a little increase in rats' food consumption when they had access to high salt, but we didn't see that. But remember, this mixed group here, shown in the purple squares, this is their total food consumption, and it doesn't differ from either the high salt only or the low salt only group. But this mixed total group, if we separate this out and just show the total food consumption for this mixed group, remember this takes into account their eating of both the low salt and their high salt diet. So we wanted to look at, well, how does consumption of just their low salt appear relative to stress? So if we look at within just the mixed group, if we hone in on their consumption of the low salt food, we see there's no differences here, indicating that they're consuming almost exclusively low salt as a part of their total food consumption. Similar here, but once we start getting after this experience of stress, learning that stressful cognitive task, that low salt food consumption is dropping down to the point where it's significantly less than their total food consumption, indicated by these purple diamonds. The red and blue diamonds indicate significant difference from low salt only consumption and high salt only consumption, which I'll bring back into the graph in a moment. But here we're seeing a reduction in the low salt consumption when they have access to both. However, their total consumption is not changing. So that shows us or tells us that when we measure, right, we see, oh, their consumption of their high salt is actually increasing proportionally. Right? So they're choosing, when they have free access to both of these, they're choosing to consume more high salt and less low salt, even though their overall food consumption stays the same. And I'm not going to go step by step through the male graphs, but when we see all the female data here, you can just see stable food consumption. But when they have free choice, right, that tells us a lot. And this is pretty important because hardly any people give rats free access to 2 different diets, or at least more than one diet, right? But this is so much more relevant to humans. Well, Melanie, are you there? Because I got a pop-up saying it. I lost a connection.
Yeah, that was strange because I didn't see you lose connection, but I did see what happened where when you click that minimize button, it sort of just kicked you out of the presentation mode. Do me a favor.
Okay.
Can you go ahead and share your
Yep.
screen again? I can edit the timeout. That won't be an issue.
Okay.
Let me see. Very strange.
It's not
Screen sharing, it's not.
I've shared it again, but it's not appearing for you. Let me try stopping again. All right, let's try this again. Whole screen. There we go. All right. Now you're
Perfect.
seeing?
I'm seeing stress
Okay.
affects female salt consumption.
Yeah. Okay. I'm not sure where I should back up to.
I would probably just start at the top of the slide again.
This one or this one?
I saw it happen on slide 24. Right now you're on 23. So
This one.
at the
Okay.
top of this one. Yep, at the top of this one.
Okay.
Perfect. And then
All right.
just as an FYI, we are at 32 minutes.
Oh, I'm so sorry.
No,
Okay.
no worries. But I just wanted to let you know because usually there is a timer, but when you're screen sharing, you're not going to see that timer. So yeah, I did want to just let you know that. And then right now, I'm just going to go on mute, and you can technically just start, like give it a second, and then you can just start when when you're ready.
Okay. I will breeze through the rest. Okay.
Okay,
Thank you.
sounds good. No problem.
So this is very meaningful and relevant by giving rats access to more than one diet and observing what they choose to eat. So what we found is that females who have this free access to both diets are choosing to eat more high and less low salt food after they experience that stressor. And in the interest of time, I'm just going to quickly show you these are the male data where essentially we're not seeing the same pattern. So what this tells us when we consider cognitive impairment as one of those indicators of neurodegenerative disease risk, we see that performance of that learned stressful cognitive task was enhanced or improved by high salt consumption in males, but not in females. And we observed that that learning process of that stressful cognitive task actually had an influence on the female's preference for salt in their diet when they could have choice. So, returning to our hypothesis, we thought we would observe reduced performance in this task as a result of high salt, but none of our data supported that. In females, we saw no effect of the dietary salt on this stressful cognitive performance. And in males, we actually observed that the high salt would enhance their long-term performance, or how well they remembered that cognitive task. So returning to this question of these early indicators of neurodegeneration risk, just want to point out that yes, diet does play an influence. Yes, stress does play an influence. And we do want to make sure that we consider biological sex. But moreover, we want to make sure that we are considering all the multiple factors that interact with each other to influence and shift degrees of neurodegeneration risk. And these are, I recognize, only 3 factors, and there's so many more out there. But I hope that this helps you understand that the contributions of salt to neurodegenerative risk isn't that straightforward. We have found through our data that biological sex is influencing these interactions between dietary salt and stress. So really, there are these very nuanced effects of the interactions between sex, stress, and salt on these markers for neurodegenerative risk that are possibly being overlooked, particularly when studies include only one biological sex when we're talking about non-human animals. So what are we going to do in the future, or what are we currently working on? Presently, we're looking at how stress and diet interact to impact endogenous hormones like aldosterone. We want to, in the future, look at how high salt intake affects cognitive flexibility, another dimension of neurodegenerative disease risk. And is this sex selective, like we've seen here? And we're just about to start studying how using DREADS to manipulate the activation or inhibition of stress-responsive neurons in the brain affects how rodents choose whether they want low or high salt foods. And with that, I want to thank you all for listening to this talk. I want to thank LabRoots for the opportunity to present today. All of the fantastic people in my lab who have made these experiments possible, our rodent caretakers, our veterinarians, my former postdoc mentor and collaborator who allowed me to pursue this project and carry it with me to my independent position, and of course funding, particularly the Ferris Family Foundation Innovator Award, as well as these additional funding sources. And if you have any questions or you want more information, this is our lab website and this is my email address. Thank you all very much.